Hello, have you ever wondered how scientists find a single molecule in a complex mixture? Today we are going to talk about HPLC or High Performance Liquid Chromatography. This is not just a simple filter, it's a super fast and highly accurate detective in the world of chemistry. Let's take a closer look at this amazing technology and see how it works. By the end of this video, HPLC will no longer be a mystery to you. So, what is HPLC? Simply put, it is a technique where a liquid mixture is pushed through a column under high pressure so that its component can be separated. Imagine you have a solution containing many different substances and you need to identify them one by one. HPLC performs that separation, identifies what is present in the mixture, and quantifies how much of each component is there. Its accuracy is so reliable that it is widely trusted for testing medicines and food qualities. There are two main players in HPLC, the mobile phase and the sanitary phase. The mobile phase is the liquid that carries the sample through the system, like a runner in a race. It is usually a mixture of water and organic solvents. The sanitary phase, on the other hand, is located inside the column. It is a solid material that tries to slow down the runners. The tug of war between these two phases determines which components come out first and which comes out later. Now let's look at the complete setup. First, there is a solvent reservoir which stores the mobile phase. Next comes the pump, the engine of the system that generates high pressure. Then comes the injector where the sample is introduced. After that, the heart of HPLC, the column where the actual separation takes place. Finally, there is the detector which identifies the separated components and displays pics of computers. Every part has specific and important role. A common question is, why do we need high pressure? The particles inside an HPLC column are extremely small, typically only 3 to 5 micrometers in size. Forcing liquid through these tiny spaces are like trying to move through a very crowded room. Therefore, powerful pumps are required to push the liquid through the column. This high pressure makes the separation process fast and efficient, which is why HPLC was originally known as high pressure liquid chromatography. Now, imagine this column in an animation. When different components enter the column, the blue particles interact less with the stationary phase, so they move through quickly and exist first. The red particles, however, interact more strongly and tend to stick to the stationary phase, causing them to move more slowly. This differences in exist times is called the retention time. The stronger the interaction, the longer the retention time. The simple principle allows HPLC to separate even very complex mixtures. Now let's talk about the chemistry involved. The most common type of HPLC is reversed phase HPLC, where the stationary phase is non-polar and the mobile phase is polar. The C18 column is the superstar in this category. It contains long carbon chains that attract and retain wavy or non-polar components. The opposite setup is called normal phase HPLC, where the stationary phase is polar. Choosing the right column depends on the type of mixture you are analyzing. The right column leads to the best result. You may have noticed that buffers are often used in the mobile phase. The purpose of a buffer is to maintain a stable pH. If the pH changes, the chemical properties of the analyte can change as well, which may affect the results. Buffer help produce sharp peaks and improve the quality of separation. Phosphate and acetate buffers are among the most commonly used because they are compatible with both the instrument and the sample. It is also important to degas and filter the mobile phase before use. As the separated components exist in the column, the detector identifies them. Different types of detectors can be used, such as a UV detector which measures light absorption, or a mass spectrometer which determines molecular weight. Once a detector recognizes all components, it sends an electrical signal to the computer. The computer then converts this signal into graphs known as chromatographs. Without a detector, HPLC would essentially be a build journey. This is the final result, the chromatograms. Each peak represents a different component. The position of the peak indicates the retention time, which helps identify the substances. The area under the peak tells us how much of that substance is present in the mixture. As you can see, the first peak belongs to the blue component that eluted quickly, while the second peak belongs to the red component that was retained longer. Once you learn to read a chromatogram, you are well on your journey to mastering chromatography. So, is HPLC always the best choice? It certainly has many advantages, including high resolution, excellent accuracy, and automation. However, it also has some drawbacks. The equipment is expensive, maintenance costs can be high, and solvents are often costly as well. Sample preparation can also take time. Nevertheless, in pharmaceutical and research laboratories, HPLC is indispensable because accuracy is of utmost importance. Also, HPLC plays an important role in our everyday lives. In pharmaceutical analysis, it is used to verify the purity of medicines. In the food and beverage industry, it helps detect vitamins, additives, and toxins. In environmental monitoring, it is used to measure pollution levels. Whenever you take a safe medication or drink a quality-tested beverage, there is a good chance that HPLC has contributed behind the scenes. This technology truly helps create a safer and healthier world. So friends, today we explored the complete journey of HPLC from pumps and columns to the detector and chromatograms. We learned how chemistry and high pressure work together to separate components, mixtures, and with remarkable precision. Hopefully, the next time you hear the term HPLC, you'll immediately think of the race between the molecules and the peaks on a chromatogram. Keep sharing knowledge and continue exploring the fascinating worlds of science with me. See you in the next video. Bye.